And we go directly to the panel A of our conference under the viral shadow. And I welcome you again, dear audience. Panel A, Bodies, Organisms and Machines, will show us current artworks from the section of art science, hybrid art and design. And it refers to both synthetic biology, but then also wearable technology studies. And we're going to embrace in our upcoming presentations and discuss interesting aspects about phenomenal dichotomies of the biological and the mechanical. And I don't want to take too much time away and want to just suggest logistically, I will present Vivian and Anna and Alex, and they will give the talks right one after the other. And then afterwards, we open up to a larger discussion. I'm very, very pleased to present Vivian Zhu, who we are very familiar with already, actually since 2014, when we prepared the non-human subjectivities, non-human agents. And Vivian Zhu is a Beijing-born interdisciplinary artist and designer and researcher. She is assistant professor of media and arts at the Duke Kunjan University. Her work explores the boundaries between bio and electronic media in creating machines and sensory systems, often taking the form of artifacts, installations, and wearables. Her work has been presented at various institutions in China, the US, Europe, and Asia, in Australia, excuse me. She has held teaching or research positions at various institutions, including Parsons New School for Design in New York, and New York University, Shanghai, and the Chinese University of Hong Kong in Shenzhen. She co-founded Dogma Lab with fellow designer, artist, and Professor Benjamin Bacon, who we will hear then in panel C. And as I said, we are very pleased to have Vivian with the amazing skin series in our exhibition. And Vivian also, we are very connected since we had the pleasure to welcome Vivian for 4 months in Berlin in 2019, together with her amazing project, the Silkworm Project. That's so much from my side. Please, Vivian, join us. Thank you for being here.
Thank you, Regina, so much for inviting me, and for you and Chris and the whole team for putting this amazing program and exhibition together. Let me just very quickly share my screen. Okay, as Regina had mentioned, I will be mainly talking about 2 installments of the skin series that I have been working on in the past couple of years. And 2 of these pieces are currently on exhibition at Art Laboratory Berlin. So my interest, I had always been interested and wanted to do something in the area of wearable technology. And I found a sort of point of entry in this skin series that I produced. One of the research questions I had while thinking about wearables is thinking about how they can be extensions or enhancements, non-invasive enhancements to our physical body, and to sort of enhance our capabilities and possibly change our perception. And one of the areas of research in biology that really inspired me is this area called sensory ecology, which, to put it very briefly, is looking at sensory systems in comparison to data and information systems, and looking at organisms as sort of these processors of that information. And so different organisms, they have different types of perception organs that allow them to perceive different slices of reality. And so animals that are maybe very different from human perception, they would experience an entirely different reality. And so I was really interested in that kind of a philosophical musing of this area, and thought, well, could we maybe map some of these animal sensory systems onto the human body, and would that then change our perception and therefore our behavior? And so, as I mentioned, this is another paper that really inspired me. An example that you can see mapped out here is, the dog and the shark, and for instance, something like a shark that is kind of a fish in underwater, because the environment that they live in is so vastly different from, say, the human environment that we're comfortable with. Therefore, they have capabilities such as sensing magnetic pull, or they have a different relationship maybe to sound and things like that, because in the water, light is not really a very prominent factor, and so whereas humans rely largely on our visual senses, and this is our dominant sense, other animals they may not, to varying degrees, and that, I think, presents this very interesting dynamic relationship through the senses between the organism and the environment. And so to really map out what I was trying to do with the project in general, or the series in general, is to think about sensory systems in the context of data systems and information systems, and to think about how data is captured and then transformed into information, and therefore then interpreted into sensory, and then how that affects behavior. And so this is really what I set out to do with some of the wearables that I wanted to design in the series. The first aspect I was really interested in was this idea of electric sensory that exists in certain animals, or sometimes in certain animals or organisms, it's just a sense of electric fields, and in some other cases it's electromagnetism. You can see here there's a pigeon that is mainly sensing sort of magnetic fields, and then there's an eel in the water, which they sense electric fields. And in some cases with these organisms, such as the eel, it's not just accepting and receiving this kind of information and data, but also having the organs to sort of project that out into the environment as well. So it's a two-way communication. And so that was really fascinating to me. Additionally, as many of you might be familiar with already, Anthony Dunn and Fiona Raby, in their book Hertzian Tales, talks about this idea of this invisible landscape of electric signals that is produced with the sort of omnipresent electronic device, and having increasing amounts of electronic devices in our lives, in our cities, in the human lived environment. That sort of invisible signaling field is actually very dense, but because biology doesn't evolve as fast as technology in a lot of ways, and therefore we as humans, our sort of biological body is really not, it cannot actually access that layer of reality that we've created, that we live in, this sort of technology built environment. And so I think, inspired by some of those ideas, I created the first of this series, the electric skin. And so the electric skin is really a wearable that strives to take some of the minute electric signals, or static electric fluctuations in the environment, and then to sort of interpret that into vibration onto the body, so that we could, through touch, feel the influence of electric fields on our body. So that really is the concept of building this suit. In reality, because I'm not an electrical engineer, developing this suit actually took many iterations and many years, and I think there are still I'm still working through trials and errors to try to get it as close to my ideal as possible. So this iteration that you see here is of the first prototype that was built in 2016. And it works on a small patch. And the idea of the, and as you can see here in the center photo is a picture of me wearing it for a photo shoot. But the concept of the skin itself is really to make small little hexagon modular units that could link up to each other. And on each unit, there are two main aspects. One is a sort of small antenna. It really relies on this sort of capacitive touch mechanism to capture any fluctuations of static electricity around it. And then there is a little control circuit around that antenna that magnifies that signal and then pulls on electricity to trigger the little motor at the bottom, at the other side of the little circuit. And so this is one little module. It was adapted from a circuit that is used to actually create these high voltage detectors that you can, if you raise it against the wall, you can sense high voltage in the walls, in the infrastructure of a building. And the idea was to then link this this unit as a module into a fabric, and this fabric could then be put onto wearables, and it could allow the human body then to sense the minute fluctuations. So the second piece I wanted to talk about that is also on exhibition is the sonic skin. So the sonic skin also looks at another sort of invisible field, which is the auditory field, and it takes inspiration from organisms such as whales and bats, where a lot of these environments that these organisms live in, as I've mentioned before, do not have a lot of light, and therefore the dominant sense is not really vision, but in their cases sonar or directional sound. And so the second piece, the sonic skin, is a prototype that creates this sort of sound wall, this wall of directional sound beams that could project from the body outwards into the environment. And the way that the sound beams are designed is that they contour the body. And so as some of you may know with directional sound, when you an example of this in everyday life would be a directional speaker. It beams sound as like a ray into the space, and you can hear it bounce around the space, and so it has this very spatial quality to it. And so by creating these different angles of sound around the body, it projects that the sort of contours of that body into the space, and the sound is a actual reflection of the relationship between the body and its and the space itself. And so this is also the concept of the sonic skin. This prototype that you see here also is an early prototype that was first created. It was prototype number 1, created in 2018. The version that is on show at Art Laboratory Berlin right now is I think version number 2 or 3. And so since I see that I have just 2 minutes left, I'll wrap this up pretty soon. So very briefly, as I mentioned, this is some of the technology research that I had referenced. These are developed by Japanese researchers, and it's really to create your own DIY directional sound. But some of this is now considered maybe not the most safest to have this kind of sound on constantly around humans or certain other animals. And so that is why in the exhibition space we turn it on and off occasionally for people to experience. I do have a sound clip here of the sound that has been played. This sound is created by Benjamin Bacon. He created it, performed it originally live in Beijing at the Second Arts and Science Exhibition in 2006 on his instrument that he developed himself, SoundSpace. So there's a link to that if you are interested. And the reason we chose this sound is because it has this sort of two-channel spatial quality to it already, and so to put it onto the suit, it would really enhance that spatial feeling of what sound feels like in space. So last but not least, this is a future third installment that I'm planning potentially for next year, which is looking at the aspect of bioluminescence and how that is used as a communication tool in nature, and hoping to develop a wearable from that that can be mapped onto the human body. So thank you so much, and I will end my talk here. Thank you.
Thank you very much, Vivian. This was really interesting. As I said, if you allow me, I would actually right away already continue with our next speaker, and I would say please collect questions and comments, and let's discuss this after the two next presentations. I have the big pleasure to present you and introduce you to very impressive artist Anna Dumitriu, who we are also very close and collaborated for more than 5-6 years. Anna Dumitriu is a British artist who works with bioart, sculpture, installation, and digital media to explore our relation to infectious diseases, synthetic biology, and robotics. She has an extensive international exhibition profile. For instance, only to name a few, her works have been presented at ZKM, Ars Electronica, Beaux-Arts, HEC, the Picasso Museum, and many, many other places and institutions. She was the 2018 president of the Science and the Arts section of the British Science Association and holds visiting research fellowships at the University of Hertfordshire, Brighton, Sussex Medical School, and Vaar Society, as well as artist in residence roles with Modernizing Medical Microbiology project at the University of Oxford, the National Collection of Type Cultures at Public Health England, the University of Natural Resources and Life Sciences in Vienna, and the Institute of Epigenetics and Stem Cells at the Helmholtz Zentrum in Munich. So you can imagine how busy Anna is. And if you allow me just to also bring up the context here in the context of Art Laboratory Berlin, we've had the pleasure in 2014 in our series of micro and macrobiologies to welcome Anna with a solo exhibition, The Bacterial Sublime. And today Anna will tell us more about her really impressive work, Engineered Antibody. Thank you, Anna, for being here.
Thank you. I'm very pleased to be presenting, and thanks for inviting me to speak and to be in the exhibition. Just going to share my screen now. So so I'm going to talk about engineered antibody and start with this to sort of, I found out from Facebook today that I was actually doing this 6 years ago. So engineered antibody started to be made 6 years ago, and I started this residency at the University of California in Irvine. And this residency started with an intensive 3-day computer-based training course, sitting in a room on my own, which if I failed it, I wouldn't have been able to actually do any hands-on work in the lab. So it was a very bizarre experience. And several of the people in the lab there had told me they'd actually failed it the first time. So I was absolutely terrified. And it's one of these things with sort of the art science collaboration, the artists, because we're on less funding and things like that, sometimes you have to be even more determined than the scientists will be. The similar thing happened with another project at Technion. But so I've fully passed the laboratory safety fundamentals, so I can also not just work with bacteria and synthetic biology, but I also know for the University of California standards to handle radioactive isotopes and all sorts of things, and fire and bizarre experience that was. So, and also hazardous waste, because I work with I create apparently a lot of hazardous waste in my work. So you can see how long ago this all was. And that's me on the sort of first day in the lab there with the special fire retardant lab coats there. They're very, very health and safety conscious, particularly so based on my experiences around the world in different labs. And that's something I really enjoy, going into these kind of different labs a lot to see the differences in how these things work. And I've written about that in a few things, because different attitudes, approaches to ethics, what you're allowed to do in certain countries, it's all very, very different. And so this is part of the world of science and understanding that. And this is sort of a kind of lab bench that I was confronted with there. And my collaborator on engineered antibody is in the photograph there, dressed up as a Chinese royal on his trip to Beijing. So I worked in the Liu Lab for Synthetic Evolution, and I worked with a scientist called Chang Li on engineered antibody, and some other scientists in the lab on various other aspects of the work. And so my first day in the lab, it's quite a few years ago, and this was my first kind of foray into synthetic biology. And they very patiently spent quite a long time explaining this to me, which is how DNA is replicated and transcribed, and how they can intervene in that by using non-canonical amino acids in it. And they made me watch videos like this about amino acids. So there are 64 amino acids, and any living thing, like normal living thing on Earth, is made up of a combination of 20 of those. It doesn't have to be the same 20, but 20 amino acids go to make a living organism. But in the lab, they're working with, can you make an organism or something like that with more amino acids in it? So they were specifically working with antibodies, for example, that rather than containing 20 amino acids would contain 21 amino acids. So this is something that wouldn't have naturally occurred, as far as we know, in nature, but something that they could create in the lab. And the benefit of being able to create an antibody with 21 amino acids is, you know, we all know now the importance of antibodies and antigens and all this sort of stuff. And if you're exposed to a virus, you need these antibodies to kind of cure viruses or treat viruses. And they, if you have a sort of a set of things that are a combination of 21 amino acids instead of 20 amino acids, you've got more things to pick from. So you've basically got more chance of finding a useful antibody that will treat a disease. And so I spent a lot of time saying, so what is an antibody and what does an antibody look like? And this is an image that they provided me with a lot, and then kind of it tried to explain things. So you've got this kind of you can see on the diagram, you've got the light chain on one side and the heavy chain on the other side, and these sort of hand kind of things that grab on to the thing that they're trying to take away from the body. So that's what they showed me. But I was like, well, what does that look like? Like, if I was trying to make one, what would that look like? And so we came up with this at this point. This is the amino acid sequence, and looped into the protein structure of the antibody in the sort of first draft of what it would actually physically be, because these antibodies and these proteins are looped, and the looping and the physics of it is what creates the function. And the amino acid sequence. And so this was the sort of first attempt at engineered antibody. And they use this bead metaphor a lot, which Regine mentioned. So they talk about it like a string of beads. And I really like this because it seemed a lot more tangible than this. So the amino acids are joined together in these chains, and then they're looped. So then I was like, so could I physically make this antibody? And so then I needed to collect the the amino acids to make it. So I wanted to go from scratch with the crystalline amino acids. So this is sort of the work in progress that I found. I was looking through old photos of the lab process, and so these are some of them. And here I haven't got the sulfur tyrosine, which was the non-canonical, the additional amino acid that they put in. But in the end, I managed to collect them all. One of them I ended up having to get out of from a vitamin shop because it was easily available as that, and they didn't actually have it in the lab. But the rest of them are all from the lab and lab supplies. So they're the amino acids, and then they work with this stuff a lot because they're saying they're trying to come up with new polymers. So I was looking at, could I create, could I use polymer clay to kind of encase these crystalline amino acids. So I ended up making this complex set of handmade beads using polymer clay, a beautiful kind of polymer clay that you can fire in the oven that goes kind of clear when you do it. So a lot of these polymer clays don't go clear, but this is a really nice one that goes clear. So the crystallized amino acids are inside them. And I had this complex process of trying to make the exact number of beads or enough beads and then keeping them all there. And it was very complicated, spread all over the floor in the house. And anyway, and then the Kumasi blue dye. This is a terrible photo of me in the studio, in my very messy studio, which is mainly boxes. This is why I never let people into the studio. People say, I want to come on a studio visit. I was like, you don't want to see what my studio's like. But anyway, so this is me in the studio where I can get very messy, working with the Kumasi blue dye, which is, it dyes everything. These lab dyes, they are so powerful. So it dyes anything with protein in it. So it's a protein based, it's a dye that dyes protein. So they use it in the lab, as Raju mentioned. It was originally a wool dye, but nowadays, because it's so good at dyeing protein, they use it for these electrophoresis gels and things, and they can cut out the little bits and you use it for dyeing DNA and all sorts of things like that. So any protein, hence the enormous gloves, because I would have blue hands for quite some time if I carried on like doing it with I'm a great believer in sticking my hands in weird dyes and things usually, but this I would have had blue hands for months. So this is it, and it doesn't dissolve in water. This stuff I was working with, it only dissolves in alcohol. So I ended up using surgical spirit to mix the dye, and this is me dyeing all the components because they use this to study the antibodies. So I thought it was a lovely medium to include, and this is the results of some of the dyeing there. So the embroidery silk, the linen. I use linen a lot as a kind of metaphor in my work, as a metaphor of biotechnology, because it's this really weird, very ancient fabric that's retted using bacteria, and it's both from plants, and it's just, the process is so complex and bizarre. It becomes this metaphor in a lot of my work, to talk about that link to history. The silk is there, also very important in the history of science. Louis Pasteur specialized in the diseases of silkworms and things. So I'm thinking of disease, I'm thinking of materiality. And this cotton calico that I dyed as well is, I think you can just about make out, representing that diagram that I showed you earlier of the antibody. This diagram that I could not get my head around because it was so abstract. And that's the interesting thing. So you talk to the scientists and they say, well, what does it really look like? And then they can't understand this because they're so used to these abstractions. It's like thinking about what it would really be like if you wanted to make it is very difficult. So I wanted to put this in there as a kind of layer. And the conversation was really strange though, because when I was like, well, I want to make it with all the amino acids and string them together and do the thing, and they're like, what? You can't possibly want to do that. I did like 452 beads. They just thought I was mad to go like to physically try and make it. It was something that they wouldn't have thought of, even considered trying to do, but I did it. And this is me stringing the beads together and looping it into the right protein structure. And actually, during this part of the process, which was online, it was an early example of an online collaboration because I could only be in the lab part of the time. We were doing this over the internet, Shangli and me, and he was like telling me how to loop it, and he realized he'd made a mistake in the representation of his protein, and we had to make some changes at that point. So it actually was useful to collaborate with the artist because physically making it actually was a benefit. And so here's some close-ups of it. And here's probably my favorite photo of it from a top-down view. It's faded a little bit, and I'm in two minds of whether to retouch the Kumasi blue or not, but I'm letting it fade a bit at the moment. And so there was a second part of it that's not on Show Art Laboratory Berlin, because the ethical rules in different countries and things, obviously this is kind of makes it more explicit. And so that piece was called Engineered Antibody. This piece I'm going to talk about was called Necklace. And I think I'm just trying to mess with curators there, because they, you know, when they're trying to do the labeling and put the labels on the wall. But I kind of, so a lot of the work with the synthetic biology is made with plasmids. So they use these tiny fragments of DNA, circular fragments of DNA, and they insert them into bacteria and yeast and things like that. And actually the plasmid in a way is kind of like a necklace. It's a circular fragment of DNA. And I kind of thought, well, it was like a necklace that the bacteria could wear inside themselves, if you think of it like that. And so what I ended up doing is working with the engineered antibody, and then inserting that engineered antibody on a plasmid into an E. coli. And I have to read this bit because it's quite hard to remember the exact bit. So what I did was I put the amino acid bead in a variable region in the antibody. So it was kind of like the process is something that occurs in sort of the natural maturation of antibodies in B lymphocytes, the white blood cells, during how it adapts to making an immune response. The B cells undergo genome editing, which is combinatorial exon selection of variable joining regions and class switching to modify and tune the antigen to recognize the site and the overall antibody functional properties. So the bacterial colonies were grown in the form of a necklace on a chromogenic X-gal plate containing a plasmid vector for the E51 light chain Fab antibody, plus or minus a single codon mutation at the position L68. This is a bit sciencey, but just in case you want to know that bit. And then each colony contains a fragment of the chain, but with a random amino acid at that position. And then the colonies grow either blue or white, depending on the compatibility of the fragment insertion into the LAC-Z selection marker. So basically, there's a random amino acid change in it. And if that change is a stop codon, then it grows one color. And if it's not, it grows another color. So you've got the exact same bacteria, but some of them are blue when grown on X-gal and some of them are white because of this one amino acid change. So you can see like a tiny thing makes a big difference. That's Felix Grün, who I collaborated on this. He really enjoyed playing about in the lab. This is me with tons of agar doing this piece. And look how happy I am being in a lab. Now, I got to go back to a lab last week, so I was back in the lab at the Helmholtz Zentrum in the Institute of Epigenetics and Stem Cells. It's the first time I've been in a lab since the whole of the pandemic, so it's been awful. But look how happy I am with this great big pipette to make the agar for these things, and learning how to bend glass loops so I could spread out the bacteria and things like that. And that's my serious face where I'm actually doing it. And this is the result. This is the necklace. So you can see, so we spread it around in a kind of slightly DNA-esque kind of thing, like that sort of natural kind of form. But it's just this beautiful change that you can see I think I really love the way that the blue and the white are just the same colonies in there, and it fades between them. So that's Necklace, which I would love to show again, but I'd have to make it from scratch because it was only shown once at the University of California in Irvine. And as with science talks, I'm going to talk about the whole team and thank the whole team there. That you can see Chang Liu and Sean Lee just hiding, peeking out there, and many of the other collaborators. So I've got some thanks to all the people, and it was a residency through the Beale Centre for Art and Technology, and the project was curated by David Vermillion and Jens Hauser as part of this wetware residency program. So that's the end of that talk. Thank you.
Thank you so much, Anna. That was really brilliant. And this is exactly why we're doing this, why we go so deep and why we do not just exhibit and why we make conferences to actually participate in your wonderful, interesting processes and artistic research. And as Vivian showed and Anna showed, that there is so much behind the piece or the work, or so we're about to try to vitalize. And so I go actually, thank you, Anna, I go right away to the third presentation, which is actually co-presentation, and Anna stays with us and Alex May comes to us. And I just realized in our project under the Viral Shadow, we have many artists or researchers couples here, including Chris and me, which is also would be a separate project to follow up. Let me please present to you Alex May. Alex is a British artist questioning how our individual and collective experiences of time and formation of memories and cultural record are mediated, expanded, and directed by contemporary technologies. His work forges creative links between art, science, and technology through a wide range of digital new media, including virtual and augmented reality, photogrammetry, algorithmic photography, interactive robotic artworks, video projection mapping, generative works, performance, and video and sound art. His international exhibition profile includes Ars Electronica, Laboral, Furtherfield, HEC, the Francis Crick Institute, only to mention a few. He holds a visiting research fellowship at the University of Hertfordshire. And here's Alex, together with Anna with us today. And thank you for being with us here as well, Alex. And so we are very pleased to get bigger insight into this interesting work, Archaea Bot. I mute myself.
Thank you, Regine. And very nice to be here with you all. Got some slides for this one. So Archaea Bot, it's a underwater robotic installation. And it is based upon these Archaea life forms, which are believed to be the oldest life form on the planet. They evolved on deep sea underwater hot vents where they're very simple unicellular organisms that just kind of sit about and absorb nutrients through their membrane. And they have this amazing property that where if they run out of nutrients, they will actually grow tails. They will spin the tails and this will propel them somewhere. They're not particularly directed, but they're not at all directed. Not at all directed. They're not like E. coli or anything. They're just kind of like wobble about, and hopefully they will land somewhere where there's the nutrients are available again. At which point the tails fall off. This is one of the major branches on the tree of life. There's archaea, there's bacteria, there's eukaryotes, which is all the nucleus based cells, ourselves, trees, plants, fish and everything. So the archaea that we're based this project on comes from this wonderful hot spring in Yellowstone Park in America, where it's discovered. It's called Sulfolobos astolcodarius. So got it right. And this was was studied by the MARA project, which Anna will talk more about.
So yeah, so we collaborated with the MARA project at Imperial College, and our wonderful collaborator, the late Amanda Wilson, on the work. And Amanda was an amazing cryomicroscopist, so she could study freezing the archaea. And I'm holding them like this, but they're microscopic, but they seem like this when you look at them under the microscope. She would freeze them in 3D and then look at them through a scanning electron microscope and try and understand the structure of these, what are called archaea motors. These little tails that they're these little motors that turn their tails that they have. And the reason the MARA project was studying them was because they were interested in talking about making them into mini robots, so sort of microscopic robotic assays. And the idea being that if they could understand the structure of these archaea motors, then they could replace the tails, they could build the motors using something called DNA origami, which is a special way that you can sculpt and fold DNA. And then they could replace the tails with kind of a DNA drill bit and use that to kind of go into the body and drill into bad cells. So I went and worked with Amanda in the lab, and this is some of the archaea being shaken around. That's why it's such a blurry photo, because they're being shaken around because they have to keep them moving, and they can grow very happily at 80 degrees. So they're very happy in hot climate. They also are very acid tolerant, as you can see from the location of the hot springs. So I was thinking, well, this is interesting. So we've got this issue of robots, you know, this discussion of robots. And it was very much when we were making this, it was very much at that time when AI was kind of considered to be the big existential threat to kind of humanity, and the letters were coming out signed by, you know, various leading scientists saying that we need to be careful about AI. So there was a lot of talk about AI as an existential threat, and this idea of the singularity and what would happen. And not only that, we were thinking about how 80 degrees, highly polluted, it could be a potential post-climate change world. And actually the archaea, they were there at the beginning, they'll be with the world, you know, at the end. If there's life on Earth, it will be archaea. They're everywhere. A lot of them are extremophiles, but actually we have archaea living inside us, they now know. And the fascinating thing with them is that they're actually more genetically similar to humans than the bacteria. So we evolved from the archaea. The way they replicate their DNA, they look just like bacteria, but they're more genetically similar to the eukaryotes in how they reproduce DNA. So that's doing some cryomicroscopy, which is one of the most hilarious things. You work with a lot of liquid nitrogen, and you get these alarms going off and telling you that there's too much of it in the air and you have to be careful. But at that point, because you're kind of breathing in this stuff, it's bizarrely hilarious because you're having your oxygen restricted. Weird moments in the lab there. And that's one of Amanda's amazing images of Sopholobus acidocardarius. And you can see these tails and these flagella there.
So we developed this project at Laboral in Gijón in northern Spain as part of the EMAC residency program there.
And with funding from Arts Council.
And funding with Arts Council England, and also working with University of Hertfordshire, Daniel Polali.
Professor of AI.
Yes. So this is part of the construction process. We'd obviously done lots of robot projects before. The particular challenge for this one was that it was underwater, and this created some specific design challenges. We also were using the latest data that from Amanda of the imaging that she was doing of the motor structures. It wasn't, you know, we couldn't get, you know, although the models were incredibly detailed, they weren't as kind of high res as maybe we needed. So we had to sort of model our own based upon these the structures that they were revealing. And this is the first kind of prototype that we had with all the electrics, motor controllers, everything inside. So it's an entirely sort of self-contained robot that is able to sense data about its environment. So it's got an accelerometer, so it can tell about how much it's moving around. It's got a gyroscope, so it can tell which direction it's facing. It's got a temperature sensor for the water, and it can also detect, obviously, which way the motors are turning and how fast. All of this data that we recorded and put through a neural network to create the control system for turning the tails on and off, which we'll talk about in a minute. So we went again through a series of interesting experiments of dunking electronic components into tanks of water, trying to find out what was the best way to waterproof them using what was that stuff called?
A sugaru.
Sugaru, which was good but quite expensive. We finally
also quite lumpy.
We finally settled on this wonderful stuff called liquid electrical tape, which was actually really difficult to find out about if you're not in the marine industry. If you're into boating, you use it for waterproofing your
Alex is now an expert in yacht electronics.
Yeah, art takes you in strange and interesting direction. And yeah, we just had these endless tests of leaving these motors running overnight. And
is it working? Is it still working the next morning?
I mean, and it's not just obviously putting metal things into water is one thing. When you pass an electrical current through them, it rapidly increases the potential for it rusting underwater. So we did lots of tests and these sort of things. Finally, we came up with this working prototype. We had a 1 month residency at Laboral, and then immediately we had to go to Ars Electronica.
It was like a 1 week gap.
Yeah, to show it. Which, if you imagine, like we've just created this first prototype, and then we put it on show at Ars Electronica. This was somewhat nerve-wracking, but we did it, and it worked
with some adjustments. That's an understatement. There may have been some soggy robots drying out in all over our hotel room. Lots of prototypes.
Minor explosions at one point, but yeah, all relatively safe. And since then we've adapted it, we've evolved it, we've redesigned the motor.
We've really got the hang of it now.
And we've exhibited at many places as HEC in
very long shows as well.
Yeah, and this is various other installations.
Bondi Marge in France, and this is in Croatia. With container, and this is actually literally under the viral shadow, because you can see she's wearing a mask over. So this was shown sort of the middle of last summer, when a lot of places were still in lockdown as well. So it was lovely to have that exhibition. They couldn't get the right size tank because of restrictions at that time, but we made it work. So that was lovely.
And then we are continuing with the project. We've got plans to develop the robot further and develop the take it into different environments. I mean, our long-term goal is to put it actually into the ocean and sort of return it back to where it lives. But in the meantime, we've just created this video artwork, where we've done this 3D rendering of Archaeabot floating around in a post-singularity world. So it's an underwater city, and this is obviously post-climate change. The sea levels have risen, and the Archaeabot are very happy because they like hot temperatures, they like acidic oceans. They're going to be fine. People have all gone, but they're just going to.
So it is, sort of thinking about how the last vestiges of human consciousness, because they've got these neural nets. We were thinking about like the uploading of consciousness, this idea that we could put our put our consciousness, it's a sort of transhumanist idea that you put our consciousnesses into digital form, and we could all live on in silica in the future or something like that. But I mean, we were thinking about how this is like a very dystopian solution, the kind of thing like a few sort of wrong thoughts from people like Elon Musk and stuff, and this might be the future for all we know. So it's kind of we describe the work as a dystopian solution for climate change, really.
So yeah, so human consciousness living on in archaea-based underwater robots. That's
yeah, so if we don't deal with climate change, maybe this is the future and we need to be careful.
And that's it. And if you can find out more about the project and the latest developments there. Thank you very much.
Thank you.
Thank you so much, Anna and Alex. That was really interesting to get that insight. And I'm very, very happy that we're gathering here and to get a deep view, talking about deep sea, getting a deep view of artistic research. So maybe now we can come all together here in the discussion round. And thank you, Vivian, Anna, and Alex, for sharing with us your interesting critical paths and travels, I would say, to actually both reveal current research, either in synthetic biology or in wearable technology or in robotics or in neural networks, together with post-ecology discussions. I would like to actually go to each of you back and have a few remarks and questions. So starting with Vivian, actually, I found this very interesting that also in your talk that you put in the center of the attention the human attention, the animal, and to sort of decenter the human, and also our sensorial capabilities that we use to move in space. So I find it interesting. Also, I have a comment on the wearable technology, but then also on the sensorial sensory ecologies. So maybe first about the wearable technology. I find this fascinating when we look into the history of wearable tech. So we actually, it starts 1500, right? That we have watches or wrist watches to wear, and over 1600 then actually for really starting watches to wear, and then calculators. So technology attached on our bodies, even calculators watch starting in the 1970s. And as I actually have seen the smart clothes and only talking about Rosalind Picard and her students in the 1990s. So actually you go and challenge that path, that I would understand it, Vivian, but then you actually briskly and really radically turn around and then also critically question wearable technology under the commercial use, and actually as if it's sort of surveillance. You use technology, wearable technologies to actually go away from humans, away from the human body, towards actually, as it turned out, also animals such as whales and bats living actually underwater, actually having not so much to deal with visual senses, but maybe rather echolocation, etc. So I find this interesting, and maybe you can go a little bit elaborate more about the term that you also used, and we also talked about sensory ecologies. I guess there's probably also a lot of the Umwelt, Umwelt evolved.
Yeah, thank you for the question, Regina. So I think for me, because I also teach, and so I in the past have developed a course on wearable technology, and so when I guide my students in thinking about this as a sort of medium, it's very simple, actually. It's putting technology on the body, right? If you dissect the two words, wearable and technology. And so then I think when you're making something, a piece of wearable technology, whether it's for commercial or artistic and experimental purposes, the question you always have to ask is, why put technology on the body, right? I think that's the first question you ask. And then the second question is how. And I think that the sort of track that I chose for this series is maybe a bit more experimental or speculative. But I find it also very interesting because I've also shown this piece in certain design fairs and that kind of a context. And when a sort of like non-art world person comes and sees it, right? They read different things into it. And I find that very fascinating. And those things that they read are not always what I intended in the design. Like, for instance, there was one person in Shanghai who looked at it and said, well, this could be used for handicapped people, right? And so I thought that was actually really interesting. It actually had nothing to do with my intentions, but they also saw some practical use in it. And so I think for me, I treat this series more like a design project than an art project, because I think sometimes one of the purposes of doing some of these projects is to communicate to larger audiences and not just audiences in certain disciplines. And so if, say, a scientist or a normal person or somebody working in healthcare looks at it and says, hey, I don't get the sort of conceptual artistic part, but I see something else in it. I think it's also interesting in bringing up conversations about wearable technology as a area.
Did you get directly, before I go on and come to Anna's engineered antibody, one more question to you, Vivian. Did you perceive it also by wearing it in a performative way? Did you get reactions by wearing it and having the sound directly as a sort of embodied echolocation? And what was the reactions of the recipient?
So I actually, for this project, because the time we didn't go into it too much, I want to develop it so that it could be worn by performers. So I myself am not a performer per se, but I did take one workshop with a black box theater artist, and so a theater performer, and so she taught me about how performance art sometimes relies on intuition and how you tap into that intuition, how you train for that, right? And so the idea is that I could develop these suits, specifically these two, in a way that they could be worn by a performer, and they can intuitively sort of react to what they experience. However, right now, this is some of the restraints also with wearable technology right now, which is, how do you make the battery smaller? How do you make something work, but also be flexible as a circuit on the body, and all those kinds of questions. And so right now, my first goal is to have the piece work, and hopefully work for a very long time throughout exhibitions. But to do that, sometimes there is a rigidity with the materials that I need to choose and all that kind of stuff, because I talked to, actually in the process of making the new iteration that's currently showing at Art Laboratory Berlin, I talked to I tried out a multitude of circuit fabricators in China, and talked to them about the different thicknesses of flex circuits, hard circuits, and all that kind of stuff, and under what circumstances they may break if they're like bent or moved around too much. And so those all create restraints for actually having it be as sort of lightweight and flexible as I would like to push it to the that level where a performer can wear it and intuitively respond.
Wow, this is fascinating. I think this is also what Alex said earlier with the IKEA, but I think all these art projects, not only from the content side of what we're discussing now, but also from the technological side, this is really revealing in its best sense, that it opens up also questions on technology, or actually also the DIY aspect is actually also important, I think, is that it reveals the technological process of the works, but then also it brings it to the surface, how things are done. And so the rawness, or the rawness of the pieces is, I think, also very, very important, also with the skinned pieces. Vivian, I would move on. I come back in a second. I would move on to Anna. And thank you so much for your insight, Anna. And with the engineered antibody, it is actually, in general, I remember back the collaboration in more than 5 years ago now in the Bacterial Sublime solo exhibition with you. It's fascinating to see how your depth of scientific research that you undergo before going into the lab, and then understanding the scientific processes, and then actually going back into a really interesting sculptural aesthetic tradition. And all this way actually is so strong, and the evolution of your works, I might say, has involves actually scientific history, medical history, things along also scientific developments, and for instance, the history of linen. So I find this always fascinating to follow the start, where you start in the lab, and then where you end up and where you go. And with the beaded necklace, it is very, very similar. I remember, for instance, also with the linen, actually that you combine the textiles and handcrafts to bridge science in the gender aspect. Let me just bring this also in here before we talk about the engineered antibody as such, because I find this very important, and it's delicate. It's maybe it's a little bit maybe layered, but I find there is a strong power of gender aspect. So great traditions of white work embroidery, practiced by women at home. So this contrasts in the era of enlightenment dominated by male scientists. And so I think even that layer is also there. And when I see maybe only secondary on the second glance, but even the engineered antibody is also actually incorporating that layer along. So I'm really fascinated. And also, the fascinating moment was for us, and that's why we definitely wanted to have also this piece shown in Berlin at some point, which we have this year now finally, is you come from the idea of beads and a necklace. And on a microbiological level, which we can only see under the microscope, which we only can really understand in the labs, then you open up that knowledge, but then you come. And please correct me if I'm wrong, but from an art historical point of view, I would say it's fascinating that necklace is now presented as if it's in a historical museum, as if it's a necklace from the 19th century. And we spotlighted it, we have black velvet in the background. So as if we come nearer and think, what is this historical object be here at Art Laboratory Berlin? And this power of this evolution actually brings us to a highly new, updated, new, actually HIV research lab. So this is actually, first of all, which I found very interesting, that the material aesthetics is how you play with this and implement them. And now you took the metaphor really by its word and made the beaded necklace. Yeah, so that I just find really, really fascinating, Anna, and that I cannot stop not thinking about material aesthetics.
Can I say something about the lab? I don't know if you noticed in the lab shot at the end when I showed you the group of collaborators, I was the only female there. So this this was still, you know, an ongoing issue with women and representation in fields of science. I look now on the website too of the lab, and it's very mixed now. So things have improved, but you'd be surprised how many labs I go into where there aren't so many women. I mean, it's improving, and then just in the time I've been doing this, I can see the improvement, but I can also see the effects. We did a robotics project in a lab recently, and we were talking about the gendering of robots, and what color do you make robots and things like that. These sort of diversity issues kind of coming up in choices about how people build robots, the sort of more humanoid robots, and we saw changes between our first visit and our second visit into what they were actually, how they were designing their robots, and those gender aspects were kind of engaged with and improved on. It's a sort of constant kind of bringing up of that.
Absolutely.
That happens a lot.
And that is actually many layers that you reveal, and this is really so fascinating. And could you maybe also go quickly into, Anna, that you said there was a learning curve even from the scientists because of your presence, because of having an artistic view into their research. Actually, could you quickly elaborate on that? That I find highly interesting, because also, you know, in art science debates, we always say, the artists take so much out of the science lab. But I think this is reciprocal, even more than we think.
Yeah, I mean, they often kind of realize things because they're having to explain things to me as well. So I pride myself in asking stupid questions and like actually having the guts to do it, because it's so easy just to nod when they're trying to explain something, and you know, and you don't understand a word, and if you don't understand one word and what that means, then the whole thing goes. And maybe they've told you a couple of times before as well, but there's so many words, kind of like new words when you're in these fields, that you have to kind of repeat and ask again. And sometimes you find that when you say, well if that works out like that, then how come that? And they go, actually that is something that we don't know, and we were thinking of doing a project on that and things. So sometimes, like, even the projects that I'm coming up with, I found this last week, they were like, well, and I'm not going to go into the details of it because it's a brand new work in progress, but they were like, well, some labs have done this and some labs have done this, so theoretically it's possible to do this, but I don't think anyone's ever tried to do both parts of this experiment to see if they actually physically work together and things. So we've kind of been developing you know, new experiments that then also tell the story of the research, and then I'll go on to make into some sort of installation or sculpture or something like that. And yeah, and with Shang Liu, it was this thing where he was going, no, it can't be like that, because it, and then just like, oh no, we need to change this, it's like this now. And then he had to go away and kind of think, and he realized he'd made a mistake in the structure of the antibody, that he thought he thought he had it sus, but when he physically tried to make it, and that's why I find, like, where like you try and understand it and then you physically try and make it, it makes a big difference. It's like doing stuff hands-on. That's why I'm so keen on doing the science hands-on and things, because you learn so much from that, and they learn as well. And then there's all these boring moments in the lab, as I like to call them, the boring moments, where the PCR machine's doing its stuff for half an hour or something, and that's when the really rich conversations come out as well, because you can ask all these like silly questions because you're just waiting, so you don't, it's not like a formal meeting or anything, you're just sort of hanging out together, and then you have these lovely rich kind of discussions, where you can kind of go off on a kind of a little tangent and a little reverie sometimes, and they'll start to go off on a kind of reverie, don't they? And that's when it gets really fascinating.
Exactly, these are these very valuable moments of radical transdisciplinarity. I mean, this is exactly what we need more, right?
I'm working on this most recent residency with Claudia Schnug, who's written an amazing book about art science collaboration, creating value for organizations. So she actually went through this and interviewed quite a few of my collaborators from the past. And so that's all as case studies in this book. And now it's really interesting because she's actually curating the thing. So she's observing whether it was really true what she heard and things like that. Interesting process at the moment.
This is really fascinating, and actually that brings me also to the Archaea bot, and also I wanted to go over to you and Alex actually, and also like respond to your fascinating presentation about this really phenomenal work, Archaea bots, and I just want to actually say the whole title again. Archaea bot, a post-singularity and post-climate change life form. I think there are so many discourses here in it, and but let me just say one thing, Alex and Anna. What I find quite charming with that piece is the play with the size ratio between the aquarium and the Archaea. The extremely oversized Archaea has a bit of a hard time and seems cramped in this aquarium, which is actually not small either. I mean, you saw one person looking at it. And so we have calculated at Roberta Berlin the size of an Archaea and the size that you produced it. So we found out that it's 120,000 times larger, the 3D Archaea that you developed. So this idea amuses me. And also the fact that the Archaea are embodied here is by means of robotics. So the newest technology meets the oldest organisms of our planet. And actually that, again, if you allow me to respond a little bit to your artwork, brings me to a theoretical moment that Chris and I had when we were writing about the biological and the machinical. And so coming to think about biological machines, because you juxtaposes this in your work so much. So that brings me to understand that the machine, basically coming from French, medieval French, or machina, it's a device basically, or from German machen. So we have apparatuses, and we could come also recent philosophy that would repropose the machine and the machinic as process, as an apparatus of ideas, ideologies, or actions. And we could add a common factor in the ideology of Kurzweil, and here we actually tackle transhumanism, and one we won't disagree with, however we much maybe disagree with Kurzweil: the biological machine, life as machine, the organisms as machine. But then actually, and this is, I think, a discussion that connects to all three artworks here in our panel. So we have, over the last 200 years, biology, biochemistry, we increased the knowledge and awareness that our bodies and minds, the result of complex processes that can be studied down to the cellular and even molecular level. And Anna just showed it with her piece. So the knowledge has been developed and evolved simultaneously with great advances in modern engineering, which in turn produced the medical and laboratory machines that allowed us to make these discoveries. So there is a conflation actually in the technological machine and the biological machine, and great fantasies of chimeras are to be expected. And so I think your ironical take of, is this a sort of a post-climate machine, or is it the dystopian idea of, how did you phrase it, the dystopian solution for climate change? I think this is exactly tackling the point of technological machines on the one hand, and as Vivian refreshingly dissented the human from her attention, the technological machine is so much our wishful thinking, human-centered in the human-centered world, whereas then the biological machine, that would probably prevail and be, as it is more complex and maybe more adaptable, would actually be, unlike the hopes of transhumanists, would probably be succeeding basically the biological over the mechanical or the technological. We can probably discuss this further. I found it very great, Alex. Thank you so much that you also showed us how that you are further developing the Archaea bots in the filmic, in the cinematic context?
Yeah, I mean, it's almost sort of part of having been through lockdown. This sort of we've had to create videos that illustrate some of the ideas that we've been working on. But this has led to its own kind of rich aesthetics and possibilities of demonstrating these ideas in a very visual form. So yeah, this new video, I mean, it's a sort of work in progress. Has a sort of somewhat chilling feeling, although to see them sort of swimming wild is quite lovely, and this is going to be premiered in an exhibition called Artificio in the Museo. Oh God,
Bolivariano.
Yeah.
De Arte Contemporaneo.
In Santa Marta in Colombia on the 20th of this month.
Mm-hmm. Really fascinating. Actually, do you have, Vivian, Anna and Alex, do you have maybe comments to one another? I see a lot of overlappings. The implementation of technology in your projects that we have seen today, or also the DIY aspect, or actually exploring biology through technology, and actually also materializing them, or giving them a sculptural aspect.
Well, I think the aspect of technology that it is an extension of our bodies, and it's an extension of our will, and an extension of our desires, from stone hand tools to the telescope to the scanning electron microscope. It's about extending our physical abilities and sensitivities, and I think this covers all of our works. And although technology gets to the point, science gets to the point where we're working at microscopic levels, and for instance, in the Archaeobot, although it's a very mechanical device, the code that controls it, the neural network that controls it, is hidden. You cannot see that, and people sort of lose track of mechanical things when they can't understand or can't see what's going on, and then that becomes hidden from them, and so it's that race beyond the practical technology that you can infer a use of into something that programmers control, or companies control, or that there are guardians of the technology, and that for one reason or another that information is kind of siloed, either because it's so advanced that general people just wouldn't understand it, or it's patent stuff, or corporate secrets, or competitive advantage, or all these things. But it's an interesting stage that we're getting to, way past the industrial sort of revolution stage.
Yeah, I really agree with, I think, everything Alex just said. I think, as you mentioned, Regina, this exhibition is full of couples. I think Benjamin and I, we talk about the Industrial Revolution and its influence with technology and the creative industries quite a lot too. And I think that also what Anna said before, there's when you're working in collaboration with scientists, there are so many words in just one sentence sometimes that you don't understand, that you have to stop and look up. We're experiencing that too. We're working with a ecologist right now, and we're ruffling through her papers, and sometimes in just three short lines, there's 6 terms we need to go and look up, and then come back with questions. But I think that process, like you said, also is I think you know, from like the interdisciplinary collaboration in itself, right? This big question of how do we actually engage in that with equal footing from different perspectives. I think that translation, I've come to understand that like translation, I think, is a very big part of it, is how do we because also some of these terms now, whether in, you know, the creative arts or sciences, they've become such catchphrases, you know. I know later there'll be a panel on AI specifically, right? And that those terms that get thrown around a lot, and but then people coming from different disciplines, you know, come and understand it in such different ways, is that really pinpointing what a term really means, I think becomes very crucial in terms of communicating and being more on equal footing. Yeah.
I really enjoyed seeing Vivian's pieces in the show, because they're just so gorgeous, and it's like this dream of us to kind of have these additional senses or to feel these ways. And I remember when I was working at Sussex University in their Centre for Computational Neuroscience and Robotics, which was one of my sort of earliest embedded residencies, they were developing something called the inactive torch, which was a kind of like bat sonar device that you could kind of, that they're experimenting with having an additional sense, and it is reminiscent of that, but taking it into this kind of much more imaginative and kind of beautiful and kind of engaging direction. So I really enjoyed seeing that.
I think this is a perfect moment also to just make to differentiate between transhumanism and posthumanism. Not that it's been confused and misunderstood here. I think we are all on the line that, for instance, the approach of the sonic skin is very near to posthumanism. I think of maybe the philosophical ideas of Rosi Braidotti to decenter the human, to actually create a reflection and an awareness of cohabitation between humans and more than humans and animals. And also that we have this I think it is maybe even 180 degrees far. And so in the opposite of posthumanism, what we have was transhumanism, which is a neoliberal desire, a purely human-oriented neoliberal desire to enhance our lives and basically use technology to actually enhance life and create ideas for eternal lives, basically. And just this as a part, I think this is very important to differentiate. We have actually now a few questions, and I would like to put the question to Vivian. So why this wearable thing cover a particularly part of the body and not cover the whole part? Can you actually give us an answer here, Vivian?
Oh, just the feasibility of wearable technology right now. So the original in my mind, originally I wanted something very light and airy, so that it would almost be like an invisible, like in the case of the electric skin, right, that it would almost be an invisible layer or like a floating golden layer of circuits over the body. That is the sci-fi, science fiction novel kind of design, right? But I think though, for me, it was very important to try to build it. Again, resonating with what Anna said, and so in building it, I think that actually more ideas come out, and the thing is also to understand actually where are we at right now with certain capabilities, and so I think the the reason it only covers parts of the body is one is due to the amount of time it takes to fabricate certain pieces, to test things, to create. So I think like if you talk about like the technology I'm referencing in developing some of these circuits, it's actually very straightforward. It's not super complicated. I'm not an electric engineer, but I think the difficulty comes in the fabrication part. So most of these things are handmade, except for things that I can't hand make, like certain things like printing circuits. And the electric skin is probably more laborious to build, because I have to solder every piece, and each little unit has 10 surface mount pieces that have to be hand soldered. I think I wrote this down because it's a complicated assembly process for the electric skin especially, and if you miss one step, then you kind of have to start all over in some cases, because the way that things go on layer by layer, and then you have to be testing each phase just for one like 10 circuit module to work. It's just I think it was like 23 steps to assemble the electric skin. And in some cases, if you say like the chest plate that is shown at our laboratory, that is a 70 module, like 70 hex piece. So that probably took just me, after the circuits were printed and everything, all the parts were bought, that probably took me a good week of scrubbing, laser cutting, gluing, soldering, testing, all that kind of stuff, just to get that one like kind of looking the way it does. And yeah, so I think that yeah, the idea is that it would cover the full body, but I think in reality it can, yeah, it's not there yet.
It's not there yet, but we are already there. We are already at the end of our panel A, actually. Time is running. Thank you so much, Vivian and Anna and Alex. We're coming to the end of our panel A. I really enjoyed it, and we got a lot of feedbacks here in the chat, but also from other sources and channels, a lot of clapping hands. And this is, maybe we have to transmit that sound at the end of the panels, not to miss that. Thank you for being with us, Vivian, Anna, and Alex. And I will now finish the panel A. But also, we are really happy to see you later on in the final discussion. I don't know if you will stay up late, Vivian, if you will be with us, that would be really cool. I think we tackled a lot of aspects here already that we will also continue to discuss in the next panels. For instance, the viral, for instance, the immune system. There were also even more questions here in the chat, and actually also the viral on the sociological level, on the biological level, on the technological level. So dear audience, thank you so much for being here. Stay with us. We're going to have a 15 minutes break now, and we will meet you again at 1 o'clock CET, 1 o'clock Berlin time, for the next upcoming panel, B, Digital and Biological Networks. And I say thank you so much for listening, and stay with us.